So as soon as I turn it on, does it allow everyone to join? Yeah, participants are coming in. All right.
Yeah, we'll give folks a few minutes to roll in.
Yeah, I probably should start that a little sooner, but it's great having you as our kickoff presentation seminar for the year. So.
Oh, is it? Oh, great.
Yeah.
Terrific.
You're the first one of 2021. So.
And is this a seminar series that you run monthly, weekly, in normal times, I guess?
It's bi-weekly, every other week.
Yeah, terrific.
Yeah.
How many people are in the systems biology center there in total?
I don't know how to quantify that exactly, but I would say on the order of, several dozens, 50, 60. If you add all the students and everyone, could be as high as 100.
Oh, wow. Terrific.
It spans a number of departments as well. So we've got groups from mathematics and engineering and basic biology and physics. I mean, it's a wide range of backgrounds, which is exciting. So.
Right.
It goes back to the initial question of like, are you a systems biologist, and who kind of classifies?
Yeah. Well, that's what you need, though. You need to bring the people, the different expertise together. That is for sure.
Yeah, and unfortunately, Vito will not be able to join us today because he has a committee meeting with a student.
Yep, no problem.
There are actually multiple people on that are at that committee meeting that are won't be able to attend, that are usual
start
attendees.
Of the year. I mean, for us right now, this week is a big admissions week. We have multiple meetings about our graduate students for next year, and organizing how that's going to work this year. So I completely understand, getting things started again at the beginning of the year is it's a big effort to spool things up again. For sure.
Well, yeah, so I think we've got a reasonable quorum to get started anyway. I'm assuming people will continue to fill in, but it's my great pleasure to be able to introduce today's speaker, Scott Dixon, who's coming to us from Stanford. And his just a brief bio for him. He got his PhD back in 2007 from University of Toronto, did a brief postdoc at University of Toronto, and where he was studying cell viability in yeast and C. elegans. And then he moved on to Columbia University to do a postdoc with Brent Stockwell. And this is really where his science took off and led to their coining the term, discovering the process and coining the term Apoptosis, this iron dependent mediated form of cell death, which is just a really exciting area of programmed cell death that I'm assuming we'll hear quite a bit about today. So, and then in 2014, he joined the Department of Biology at University of Stanford as an assistant professor, and is now an associate professor there. And he's continuing to do amazing work on studying the dynamics and understanding the molecular machinery regulating this new form of programmed cell death, and probably broader forms of programmed cell death as well. So thank you so much for being here today, and take it away.
Thank you, Darren. And it's really my pleasure and honor to be here. And as you mentioned, to be the first speaker of the year. That's wonderful. And so thanks so much for the invitation. To your point,
let me do a quick reminder to everyone that because you cannot speak up directly, if anyone has any questions, please post them in the Q&A portion down in the bottom there, and Kavya or I will try to make sure Scott's aware of those in case he can't see them. So thank you.
Right. Yeah. And they do disappear, and I have to close some things so I can see the screen. So yeah, just let me know if anything pops up. And so actually today, what I thought I'd talk about has nothing to do with apoptosis. So you will actually hear the other sort of part of my lab, but I think it's really relevant to a lot of the work that's being done there in the Systems Biology Center. So I'm excited to talk about it and to get your feedback and to discuss it. And I'll just say up front that this is work that I'll be talking about done primarily by a graduate student, Giovanni, as well as a former PhD student, Zintis Indy in the lab. And we've had some excellent collaborators here. And we've been grateful for the support of the NIH as well as some outside funding from some biotech companies. So today, what I'll talk about in this seminar are two things. First, our efforts to quantify cell death at a large scale and do this systematically. And then secondly, how we've realized, we think, that we can use some of these technologies to study this process of fractional killing in cancer cells and try to derive some insights. But as you'll see, it's a large area and this is a little bit tricky. So I'll start by talking about the quantification of cell death at scale. And so my lab takes a very chemistry-first approach to understanding cell death. There's a whole world of molecules, natural products and synthetic molecules, many of which have interesting bioactivities. Many of those have activities against cancer and can kill cancer cells in different interesting ways. And so the general approach that we take when thinking about our science is that we want to identify lethal molecules, we want to determine the lethal mechanism, and then we want to translate that in some way to the in vivo context. And so this has been really the guiding principle for my lab for the last 7 years. To do these first two things, to identify the lethal molecules and understand their mechanism, of course, you need methods to accurately quantify cell death. That's what we want to study. We want to study cell death. And if you're screening molecules, you need to be able to quantify cell death in a large scale way. And so one of the issues that's emerged as people have moved in this direction is that it's very hard to do this. Many of the assays that existed, especially when I was a postdoc a few years ago, to quantify cell death in a large scale manner, and by that I mean, hundreds, thousands, tens of thousands of samples in a single experiment, are very indirect. And for that reason, they have a number of confounds that lead to discrepancies between large studies. And our feeling was, and my feeling certainly when I was using them, can lead to discrepancies in understanding cell death. So when I started my lab, one of my major goals was to develop some better ways to study cell death, to actually measure that process. And I'll just point out, of course, we're not alone in these efforts. And so this paper here from Vito and Darren there was very influential in our thinking about how to go about doing this on the development of the DIP method, among other work, of course, from various groups. So here's the problem when you want to quantify cell death in adherent cells. The classic way to do this would be to use some type of traditional microscopy where you can put a plate into this incubated chamber, and then you can follow a bunch of markers and look at the cells directly. And it's a wonderful method because it's very direct. You get very rich phenotypic information about how the cells are dying. You can look at the morphology, and also you can set it up to look at the cell death over time. So you get information on how that process proceeds. The con here is that it's extremely low throughput. Even with the best sorts of setups like this, you might be able to look at 4 or 5 different conditions in one experiment, and you might, be able to look at a couple dozen cells in each of those conditions. So it's not especially suitable for doing large-scale studies. I mean, even doing something as simple as a compound dose response series would be, a lot using this approach. So what do we do? Instead, what we typically would do is turn to various fluorimetry-based methods. And one example would be cell titer glow. Another one would be, Alamar blue, both of which we use and have used in the past, where you have a population of cells in a well from a microtiter plate. You lyse those cells, and then you liberate some ATP, which is then used to generate a light signal. And the pros of this approach, so then I'll just say you get the signal, and then you can generate some sort of percent viability score where you look to see the relative difference between a control and some different conditions. So the pros of this naturally are that it's extremely high throughput. You can set up 100 plates if you wanted to, lyse them all and get a cell titer glow value from them all in a day. And it's relatively cheap. I mean, when you do large scale, nothing's cheap, but it's relatively cheap. The cons are that you have absolutely no idea what's really going on, I would argue. You don't know whether these cells, for example, in this drug A condition are just not proliferating. Have they arrested? Are they actually dying? Is it some mixture of those two? Is their metabolism altered? And that's why you get a weird readout on an ATP-based assay. you also don't know what the cells look like. Do they have any interesting morphology that might give you an insight into how they're dying? Since, as Darren mentioned very briefly, we now know that cells can die through a couple different cell death pathways. The other thing that was apparent to me as a postdoc and really motivated a lot of my thinking in wanting to develop a new method was that if I was doing an assay and I took my measurement at 24 hours or 48 hours or 72 hours for the same condition, I noticed I'd often get different results. So at 24 hours, I might see that neither of my drugs are lethal. At 48 hours, one of them might be. And then if I waited another day to 72 hours, they both were. So depending on where I collected the data, I could get different results and yield different interpretations. And that, for me, was a troublesome realization. Okay, so what we wanted was a way to analyze cell death more directly and to do it over time or kinetically. And so the approach that we came up with was to take any given cell of interest and introduce transgenic expression of a nuclear localized MK2 protein. So now the nucleus is marked by the fluorescent protein expression. It's very easy to segment that by imaging. We can incubate these cells in multi-well plates and we can see the nuclei. We can also add cytox green, which is a dye that normally doesn't enter the cell until the membrane is permeabilized and the cells are dying. And then we can see this transition when we add something that kills the cells from a live cell with a red nucleus to a dead cell with a green nucleus. And here is just a video showing you sped up what this would look like over time. Here is the start of an experiment. We have a field of cells, all of which express this red nuclear protein. And then we have added a lethal compound. And now over the course of a day and a half or so, many of the cells have died. They've taken up the green dead cell marker dye. And it turns out that this is quite easy to quantify the live and dead cells over time in an automated fashion. So you get data that looks something like this. You have time here on the x-axis, the number of objects on the y-axis. And here in red are the live cells. And then in blue, I'm depicting the dead cells. And so you add the drug at time 0, and then you make these measurements repeatedly over time. You image the same population, and so you can see that the cells increase, actually. Typically, they don't die instantaneously in most cases. Then they start to die. Let me see if I can get my laser pointer going. And then you see an increase also in the number of dead cells, and then you see this technical loss of cytox green staining as the cells disintegrate over time. So this is great. It's wonderful to be able to directly measure this. But of course, now we've created a bit of a new problem. Before we had one value to represent your entire population, and now we have potentially dozens or hundreds of measurements. So we had a new challenge, how to summarize these kinetic data. And so the approach we took was to integrate the live and dead cell counts into something we call the lethal fraction. And you might hear me refer to this, where 0 represents everything being alive in your whole population, and 1 represents everything being dead. And so that's good. You've reduced the dimensionality a little bit there. You're down to 1 number, and then you can summarize that further. But what we realized was even more valuable was to take these lethal fraction curves over time and then fit them with a mathematical model shown below, where it allowed us to parameterize these curves. And it turned out that this model fits a wide variety of different cell death kinetic events, you might say, or cell death treatments exceptionally well. And from this model, when we fit it to the lethal fraction curves over time, we can then extract 2 key parameter values: the length of time between the exposure initially to a lethal stimulus and when the cells in the population start to die, and we call that the death onset or the lag phase, and then the maximal rate of that death. And as I'll show you in a minute, it turns out that those vary quite a bit between conditions, which is not somewhat intuitive, but also not something that people have really quantified in great detail before a few years ago. Okay, and so we can set up this system with multi-well plates in an IncuSite microscope that lives inside a tissue culture incubator. And we've, we were told not to do this, but we did it and it was fine. We can do, up to 6 plates simultaneously and image every 2 hours, such that we can collect about 2,000 different conditions at a time in one experiment. So it's a great way to, generate this large scale cell death data, but have it be more direct and be able to look at the actual cells themselves. And so we called this approach scalable time-lapse analysis of cell death kinetics, just because we needed something shorter than that, a stack, and you might hear me refer to that during the talk. So having developed Stack, there was a number of questions that we wanted to try to address with this technology. And so I'll just sort of step through a couple of those questions, and I'll show you some of the examples of the data for some of these questions. So these are often very simple. So do cell death kinetics quantified using STAC vary between lethal compounds? And the answer is yes. And do they vary between cell lines for the same compound? So does the genetic background of the cell, does that influence the kinetics and the way the cell die? And in both cases, the answer turned out to be yes, so far as we've been able to tell. And so let me show you and illustrate the method a little bit more with some results here. So we had first a goal of identifying compounds from a library that were lethal. So we took 2 cell lines that were very genetically distinct, T98G, these are glioblastoma cell lines, and U2S, these are osteosarcoma cell lines. And we exposed both of them to a library of 1,833 different bioactive compounds at 1 dose in a single experiment, and then imaged them over time using the STAC method, and then computed all those metrics that I mentioned. So from them, or from this experiment, we identified a total of 80 molecules that were lethal at some sort of arbitrary threshold to both the cell lines, as well as 60 compounds that were exclusively lethal to one or the other. But we focused mostly on the 80 compounds that were lethal in common. And so let me show you what some of these kinetic data look like for these compounds between cell lines. So here, what I'm showing on the x-axis are the timings of the onset of cell death. So that lag phase between adding a compound and when the cells start to die. And then here on the y is the maximal rate of the death. Oh, something popped up here. Let me see. Is this a question? Yes, it's a chat.
It is. You got
Should
it.
I answer it now?
Yeah, that's great.
Okay. All right.
Do you want me to read the question?
I got it here. I think I'll read it out and we'll see if this works. Okay. Is that model generically a good fit to the data? It looks like the model is flatter at the top of the data, at least in the pic you showed. Are there good theoretical reasons for choosing that one? It looks like it's being, okay, let me answer the first part first. And let me make a general proviso here. And we touched on this before, I think the seminar started. Not being a mathematician would be the key one. So we tried fitting a large volume of sample data with a large variety of different curves. And it turned out that this one that we ended up choosing fit the data with the smallest residuals in the largest number of cases. There are instances where, of course, any model is not going to fit a particular set of data. One example here would be if you add directly to cells a detergent that permeabilizes the membrane instantaneously, where you go from 0 death to 100% death, in the first time step. This model has trouble with that. It also has trouble if there's sort of low levels of cell death, and it can't quite identify what the lag phase is. But in our experience, for most well-behaved compounds and typical cell death processes, the curve seems to fit the data quite well. And intuitively, what we're seeing with the extracted parameter values matches, our sense of what's occurring in the biology itself when we look at the cells, which maybe is the most important thing. But I think it's fair to say that not being specialists in model building, that there really is an opportunity to do some more systematic analysis of different curves and see if it's possible that there are extreme or outlier cases where you might actually benefit from employing a different type of curve. I hope that answers that part of the question. I guess you can let me know in the chat. Second part of the question is, it looks like it's being modeled as a simple birth-death process. That's a good question. We're not doing that explicitly, and I will be honest, I'm not sure how you model a birth-death process, but if that's something that when you look at the data makes sense, you should probably explain to me after how you do that modeling. I'd be very interested to know. Okay. Oh, and then Darren's just saying, right. Okay, we've got it in the Q&A tab. Okay. I'm going to carry on then. I hope that addressed the question, and thank you. Okay, so back to the slides. So we had these 80 compounds that were lethal. We wanted to understand whether the kinetics varied between compounds and between cell lines. So I'm showing you here, each of these dots is a single proteasome inhibitor. So in this large library of compounds, we had actually multiple different proteasome inhibitors, 9 in total. We had, bortezomib, carfilzomib, and so on and so forth, all these different structurally distinct proteasome inhibitors, which we could then use as a set and say, okay, do all the compounds that target the same thing behave similarly? And in this experiment, it looks quite good. And what you can see here is that they initiate cell death with an onset of roughly 20 hours in this particular rate. And it's essentially similar in both of these cell lines. Okay. However, we started to see differences in other classes. So for example, if you looked at a set of HSP90 inhibitors, of which I believe there was 8 in our library, now you can note 3 things maybe that are of interest. First, the mean onset is much later for HSP90 inhibitors than for proteasome inhibitors. So for whatever reason, proteasome inhibitors can initiate cell death within 20 hours or so, and for HSP90 inhibitors, on average, it takes a little bit longer, over 30 hours. Secondly, you can see the maximum rate is much lower, and we were using concentrations of these inhibitors that are quite far above their published EC50 values. So we don't think that this is simply due to incomplete target inhibition. We think that this is actually due to differences in the way cells respond to these different stresses. And then third, you can start to see now a separation between the 2 cell lines where T98G cells are starting to die a little bit earlier than U2OS cells on average in response to the same inhibitors. On the other hand, we saw a different pattern with topoisomerase II inhibitors, where now what we could see was that the T9G cells were dying with a little bit of a slower onset, and the U2OS cells were dying with an earlier onset. So it wasn't as if one cell line was just generally more sensitive, but rather there are these seemingly stable differences in the kinetics of cell death within a population that we can detect quite easily now using this stack method. And so the underlying biology for these differences is unknown, but of course the first step here is to be able to quantify these differences, and now we can study them, and this is something we're interested in to try to understand, for example, why is it that proteasome inhibitors induce apoptosis with very rapid rate compared to HSP90 or topoisomerase inhibitors, which induce apoptosis through the same pathway terminally, but with a much slower rate in the population, and we think that's an interesting problem. Okay, so the kinetics vary between compounds, between cells, across lethal compound concentrations, as you would assume. They vary between cell death pathways. So if you induce apoptosis or you induce ferroptosis or other types of non-apoptotic cell death, they have unique kinetics. But it turns out you can actually find quite a broad range of apoptotic kinetics and a broad range of ferroptotic kinetics. So it's not distinguishing. You can't say just based on kinetics, oh, this is non-apoptotic cell death. And then do the kinetics vary between individual cells within a population? And they do, and it turns out that this has been an interesting problem for us to get into and try to understand in our more recent work. And so I want to spend sort of the bulk of the seminar discussing some of these more recent findings here. I see a question. Okay, let me get to that then. Okay, from Laura. Do you think at all about the induction of senescence or quiescence rather than just death? Very nice question. Thank you. I do think about it. And one of the nice things about the Stack platform, I guess there's 2 things that are useful here. One, because we're measuring live and dead cells, we can very easily identify conditions where there's no death, but there is an arrest of growth, right? This is one of my frustrations with indirect methods. You couldn't tell if you saw by Presto Blue or Alamar Blue a viability reduction of 50%, whether the cells were dying or whether they were arrested. When you're measuring, actual live cell counts, this is trivial, right? It's the same as in the DIP method developed by, Darren and Vito there. You just have a direct measure of whether the cells are proliferating or not. And so we can see that. Whether they're senescing formally, and expressing like senescence-associated beta-galactosidase and other markers, we haven't gone that far, but we certainly see cases where compounds will result in these massively large and what looks to be endo-reduplicated nuclei cells. I assume they're senescent. It could be studied. We haven't studied it ourselves in great detail. Okay. John,
are you going to follow up? So your last box there, you talk about the individual cells. So you just brought up an excellent point about, well, answering that question, you talked about seeing some cells in this senescent-like phenotype, whether it's carrier or not, but are you going to follow in depth about this part, the individual cells within the population? Because that's where a lot of the excitement is.
Right. Sure. Then that's exactly what I'm going to talk about here in some detail. Okay. But I see John has another comment with an equation. That's going to scare me. But you know what, John,
John he'll be available after in the discussion.
Let's talk about that after. I think I'm not going to be able
perfect time for that.
Yeah, to consider that very clearly. But yeah, I want to move into the second part, thinking about this issue of fractional killing, which is really linked to differences between individual cells. Okay, and so the underlying concept here, which I'm sure many of you there are familiar with, is that this clinical observation going back decades by oncologists, that if they treated a patient with a drug in the clinic for cancer, many of the drugs were typically killing a constant fraction of cells in each round of treatment, not necessarily a constant number. So if you started with 100 billion cells, in the first round of treatment, you might kill 90 billion. And then at some point you get down to 100 million and you don't kill 90 billion again, of course, you only kill 90 million of them. In each round, you're left with some small subset that remain viable. And, maybe 1 in 10,000, maybe less, maybe more, depending on the disease and maybe depending on the treatment. But it seemed that the impression created was that you're killing a fraction of the cells each time, not a constant number. And so what is the basis for this? Why is it that some cells consistently seem to evade therapy and then seem able to regrow and give rise to cells that once again can respond mostly within a subset that are resistant? And so this is actually a figure from one of Vito's papers, which I just love, because it illustrates this idea through this schematic here, where you have cells in orange, green, and blue in different states. And the idea is that they're constantly transitioning between these states. And so the argument in this 2018 paper is that it's epigenetic states, but I also can imagine, it could be epigenetic, it could be metabolic. The key point is that genomically, they're relatively the same, but they can hop between these different states for various reasons within a population that's otherwise mostly isogenic, and have different phenotypic responses. And so in response to a drug, you might select for cells to enter one particular state. And then if you take the drug off, they're going to repopulate these alternative states. And so it might be this one particular state that allows, for the survival of the small number of cells in cancer observed during fractional killing. But this idea that there's multiple states in a population, is very intriguing. And what is the basis of that? A really trivial observation, of course, when you think about it, and it's so trivial, we do it in the lab every day, but the fact that you can compute an EC50 value for any given small molecule where you're killing half the cells and not killing the other half at a given time, immediately suggests that there's some differences between the cells that you're working with, right? They are responding in different ways. No two cells are identical. And so this is a really fundamental problem in biology. It has, implications for all sorts of different things. And we wanted to get a toehold in this area and really think about what this meant. So in the context of, okay, before I launch into that, I see there's another chat. Do you want to ask a question, Bishal?
Bishal's raising his hand. I'm trying to
Oh,
ask
okay.
him if he is
He wants
asking
to-
a question
okay.
or whether there's a comment.
Sure. No. Okay,
That-
no problem. Okay, so
Okay.
I'll carry on. Okay, so what accounts for fractional killing? And I think there's a couple different models, out there, and I'll just highlight a couple of the ones I think that are quite, that we think about quite a bit. One is this idea that there's drug-tolerant persisters within a population. This goes back to work in bacteriology from the 50s. Where you might have a small number of cells that are in an altered epigenetic state, and for that reason, they can survive the treatment with the drug, and then ultimately even maybe give rise to a new population. And so that's work from Jeff Settleman's lab originally in 2010, and then there's been a lot of recent developments there, of course. Then two other models, one from Peter Sorger's lab relates to the actual apoptosis machinery, that maybe the cells have differences in the expression of different apoptotic effectors. And so depending on the levels of those effectors, you get different timings of caspase activation. And so that yields different fractions of cells killed under any different condition. And then another idea is that the upregulation of certain proteins, in particular P53, as Galit Lahav's lab has suggested here in this 2016 paper, that might be the key determinant. So if you are able to sense a lethal stimulus and you rapidly upregulate P53, that's going to be pro-death, whereas if you upregulate it more slowly in blue, that's going to be pro-survival. And so there's a couple different models here about how you might get these differences in a population, and whether one model is correct, all the models are correct, whether there's interaction, I think it's not entirely clear, and that'll be an interesting discussion to have. One limitation here is that many of these studies trying to look at fractional killing, of course, you need to do the kinetic analysis and you need to look at the cells. So you're stuck in that zone of using microscopy under low throughput conditions. And we wondered, maybe with this new stack method where we can look at, a relatively large number of conditions, we'd be able to, have a larger scale, a bigger analysis of what fractional killing is and maybe gain some insights. And so that's what we wanted to focus on and where we thought we could make a unique contribution. So let's use our methods to start investigating fractional killing under some different conditions. And so here is just some data, again, to illustrate the point that no two lethal stimuli are the same, and they often vary in interesting ways. So these are, again, two cell lines, T98G in the top, U2S in the bottom, time is on the x-axis, and then we're looking at lethal fraction, which again is our metric where you go from 0 to 1, which means you have no death at 0, and then the population's fully dead at 1. Here are 4 different molecules. ML162 is an inducer of ferroptosis, so not even apoptosis. Etoposide, thapsigargan, paclitaxel, these are classic inducers of apoptosis. What you can see by looking at these curves is that they have very different kinetics, and they have different kinetics between the cell lines. So, there's some folks in the fractional killing fields like to focus on what I would say is the EC50 situation. So where you find a dose that kills half your cells under a given scenario, and then what is it that allows half those cells to die and half the cells to survive? And that's a really important area to be working in. But what we sort of realized quite quickly is that the EC50 condition, you might say, is not stable in any way. You can identify conditions where you're killing half your cells, but that might only be a transient point in time. It might only be a limited range of doses. And really, if you wait long enough, kinetically, you can kill essentially everything within a population. There might be a few small cells remaining, but you can kill the vast majority of cells. Whether these remaining cells are persisters or the idea, that I really like of idlers proposed there in your department is really intriguing, right? There might be some small subset of epigenetically distinct cells that just resist the death, and that accounts for the 1% to 2%. And I think that's entirely reasonable. And I know there's a lot of interest in those small subsets. What attracted our attention, though, and where we thought we could have a different contribution, was in understanding this difference here in the bulk of the population. So what we're showing here is that there's some cells that are dying in these populations, let's say within 24 hours. And then in the same population, there's some cells that don't die until 2 days later, until 72 hours. And what is that all about? Why do you get these large variations in death within the bulk of the population? And I think I would argue that persister theory or idler theory doesn't necessarily speak to this directly, right? It's more concerned with those rare cells as far as I understand it. We were interested in this bulk sort of difference between cells in the population. And it didn't seem like the persister theory or a distinct epigenetic state could account for this. Now, of course, there could be graded differences. But when I think of persisters, I think of something that's in a unique state that's sort of a different phenotype altogether. We wanted to understand this sort of bulk variability. And so how variable is this really across conditions? This is something we thought we could address with the stack measurements that we've been making. And so here I'm showing you some results from U2OS cells. This is a cancer cell line treated with 139 compounds. And this is just one trace for every different compound that we examined over time. Again, the lethal fraction here on the y-axis. And you can see there's quite a bit of variability in the kinetics of death. So how the cells are dying in the population. When we plot our kinetic parameters, and in particular, when we plotted our rate, our maximal rate of death, what we saw was that most compounds clustered typically in a very low range with a couple outliers. So this one outlier here directly permeabilizes the membrane, and that's this curve that I mentioned here. But mostly, they have relatively, I would say, low rates. We like to think of them, in terms of percent cells in the population dying per hour. And so the median here is only 2.4. So when you think about that, for any given stimulus here in this set, you're only killing 2.5% or so of your cells, let's say, per hour. That is kind of interesting. I think you could have argued a priori that maybe you'd actually be killing 10% or 50% of your cells once death started, that all the cells would be equal. But this to us suggested that the cells were actually quite different. And so it was remarkably high variability we felt, and we wanted to understand this. We also noticed, again, this is similar to what I mentioned earlier, that this rate or the speed of the death in the population varied consistently between compound classes. So proteasome inhibitors are relatively fast lethal molecules in the sense that more of the cells die more synchronously, whereas we noticed that inhibitors of the mitogen activated protein kinase, MEK1/2, were very slow, meaning that differences between cells in the population were large. So one cell would die, then there'd be a number of hours pass, and then another cell would die, and it would be more asynchronous. And so we thought this would be a good area to focus on. what is going on here with these MEK inhibitors? They have slow kinetics. They're both being advanced into the clinic for cancer therapy and the subject of intensive investigation as monotherapies and also as combination therapies. And there's great tools available. So we used pimasertib and trametinib. Trametinib is something that I think is in the clinic now. Pimasertib didn't quite make it, or maybe has been superseded by these more potent MEK inhibitors. And then there's also direct inhibitors of ERK1/2, the downstream kinase. So this is a RAS MAP kinase cascade. And we could use all of these tool compounds to confirm whether the observations we're making were relevant to the whole pathway or just to MEK1/2 and so on. So here is just some summary data presented as a heat map where the lethal fraction is now color coded from 0 in red to 1 in purple. Doses are here of pimasertib and of trametinib. And all I wanted to illustrate is that you can see dose-response relationships over concentrations. You can see effects over time, but ultimately you're able to kill most of the cells at certain doses. So 10 micromoles of pimasertib or a larger range of doses for trametinib. But in both cases, for the first 48 hours or so, the cells just don't die in this condition. And then when they do start to die, it's quite slow. So what could be explaining this very slow kinetics or this slow fractional killing in this MEK inhibitor induced and treated cells? Well, we started really with all the stuff that I mentioned earlier, the mechanisms that have been proposed by other investigators to account for fractional killing. So the first one was was it perhaps p53 levels? Maybe it was the rate of p53 induction. And I'll just tell you, I won't show you, but we fiddled around with p53 levels, both overexpressing it and stabilizing it and deleting it. And that had no effect on the kinetics, at least for MEK inhibitor induced fractional killing. An obvious thought is that maybe it's differences in the cell cycle phase, either when the compounds experience the drug or as they enter the cell cycle after experiencing the drug. And we did some very careful tracing of individual cell lineages with drug addition, where we followed the fate of the cells before or after. And really there was no connection that we could see, no clear correlation between the cell cycle phase of the cells when they experienced the drug and whether they died and with what speed. There's a whole other body of literature that cells that can evade therapy might have differences in metabolism. And in particular, they might accumulate low levels of ROS, or reactive oxygen species, and that protects them in some way and prevents cell death. But we looked at this, we saw no evidence that ROS were being accumulated differentially between cells that lived or died. And when we fiddled with the ROS levels by blocking the accumulation or increasing the accumulation, it had no effect. Okay, we've done other metabolic measurements too between cells that seem to be able to survive longer and cells that die earlier, and we really haven't found anything that's striking in the metabolic space between these different populations. A trivial explanation would simply be target engagement. Some cells you inhibit the pathway more fully than other cells, and we looked at that, of course, carefully, and we really don't see differences there either across doses or over time. So here's just some actual results now with pimasertib and trametinib at different doses, and I recognize this is bulk Western blotting, but what we noticed was that even by 2 hours across pimasertib and trametinib concentrations, you're completely blocking to detectable levels the phosphorylation of ERK1 and 2. We also see this with a single cell reporter assay. You recover some degree of ERK phosphorylation at these non-lethal doses, but at a lethal dose, 5 micromole, it's maintained at a fairly suppressed state. And then here with trametinib at these lethal doses, ERK is suppressed still 48 hours later. So we think that we're blocking the pathway quite effectively, and that it's not being reactivated to an appreciable level that would, again, explain how you get this wide variability where 10% of your cells might be dead by 48 hours, but then the last 10% don't die for another 3 or 4 days.
Scott, can I ask a follow-up question about the cell cycle phase?
Sure.
Was that only at the cell cycle position at the time of treatment, or did you look to see? I mean, I would expect with the MEK inhibitors in particular that the G1 times would actually expand fairly substantially. So I would suspect, this is just pure speculation, but I would suspect more death during the G1 phase because the G1 times are so much longer. Did you look at that at all?
I can say 2 things about that. One, I agree completely. The MEK inhibitors, in most cell lines where they're not lethal, they arrest the cells. And we've seen cyclin D downregulation as well as all the other things. They definitely arrest. The experiment that we did was to take unsynchronized cells in the population using a different microscope setup, and then start following them for about 10 hours, and then add the drug, and then continue to follow them for another 2 days, and look very carefully at when they divided and then which ones died and so on. And so we didn't have a marker of G1 arrest necessarily. We were relying on morphology of these cells. But what I can say is that the cells, we tried to then line up when the cells, divided, when they experienced the drug and when they died, and we couldn't find any relationship between those. So there's cells that would divide and then die right away. There's cells that had divided hours ago before they saw the drug and they would live. We just couldn't see a pattern. But I'll admit we only looked, I think, at 100 cells, and maybe we need larger numbers. I'm not sure. It wasn't obvious that there was a real difference though with cell cycle and when the cells actually died. The arrest, that could certainly have varied between them, but when they actually died, there was no consistent pattern.
Great, thanks.
Yeah. Oh, one other comment I can say actually is that we forced cells to arrest with p53 overexpression. So we drove them into an arrested state. Then we added the MEK inhibitors and they still died with the same slow kinetics. Okay. All right. So, an obvious explanation would be, well, there's some population of resistant cells, and we excluded that, of course, by doing the classic sort of experiments. We killed most of the cells, took the drug off, let them regrow, and then killed them again. Okay. So, right. So one prediction from this study, from our approach is that if we modulate the apoptosis pathway, we're going to shift the kinetics of fractional killing. So we thought that, if you have a population that has really high heterogeneity in some way and a low rate of death, we might be able to shift that to the left, you might say, and increase the rate of death and reduce the amount of heterogeneity. And that would be a means of demonstrating that a particular manipulation was important for the rate of killing or the rate of the fractional killing in that population over time. And so if we block an important pathway, we felt we should shift this curve. And so given what was known about the MAP kinase pathway and apoptosis, and given the work of the Sorger lab and others looking at the relationship between apoptosis and fractional killing, we started there. And so again, we took really a pharmacologically driven approach using a number of different inhibitors of the pathway where we could block selectively BCL2, BCL-XL, or MCL1. And then we would perform these experiments in the presence of our MEK inhibitors and in the presence of these inhibitors of these anti-apoptotic BH3 only proteins. So the first thing we thought was that BCL-XL was going to be the answer because that's been known to regulate MEK inhibitor-induced apoptosis. We certainly saw that when we inhibited BCL-XL that we could accelerate the onset of death in the population. So from the gray curve to these pink and purplish curves. However, when we quantified the kinetics, we were only shifting the onset of death. We weren't accelerating the rate of death, which to us suggested that the population was still heterogeneous with respect to whatever the mechanism is that yields that heterogeneity. Yeah, it was still heterogeneous. And we were just biasing the likelihood that the cells would die, but we weren't getting rid of whatever it was that was allowing the cells to be really different from one another. On the other hand, when we did the same experiment with pimasertib and trametinib and inhibited now MCL1, we saw that we were getting this expected acceleration in the rate, so the steepening of the curve, and we could quantify that here on the right-hand side. And so we thought that this was an important insight into what could be regulating the heterogeneity between these cells in response to the MEK inhibitors, is that if you take out MCL1 from the equation, essentially, you're going to have a population of cells that is more equal with respect to their response to MEK inhibitors and dies more synchronously, with fractional killing appearing to occur more quickly. And so that was a hint that it was a key player. What we also have found is that the regulation of fractional killing is very variable. So it varies between cell lines, and we've looked at many because that's easy for us to do. It varies between lethal molecules. So here's a good example. We also have in this experiment bortezomib. We can see that in the case of bortezomib, inhibiting MCL1 also shifts the onset of the death, but it has no effect on the heterogeneity within the population. And so one conclusion we draw from this and some other experiments is that there's a variety of heterogeneity generating mechanisms that are going to be specific for different lethal stimuli. And that's a little bit daunting when you think about the applications. It's a little bit daunting when you want to try to develop a unified theory for fractional killing, but I think that that's just going to be the reality, that heterogeneity in many portions of the intracellular network can give rise to heterogeneity in the cell death responses, depending on how you're trying to kill the cell. Okay, so MCL inhibition makes cells more similar with respect to the kinetics. And so then we tried to quantify this at single cell level by microscopy. And I admit that, we haven't gone as far here as I think we want to go in the future. But what we've done so far is to image the abundance of MCL1 in different individual cells. And so some observations here that we made were that MCL1 is quite heterogeneous between cells. So this is now a normalized intensity of the MCL1 signal, which we infer reflects the amount of MCL1 in the population. And so in response to pimasertib, BCL-XL levels sort of collapse down. They actually reduce overall, BCL-XL is lost, and all the cells become more similar in their levels of BCL-XL. MCL1 is a bit of a different story. They're not any more heterogeneous to begin with, but they maintain that heterogeneity in response to pimasertib MEK inhibitor treatment, and it doesn't collapse down. And in fact, MCL1 levels increase in response to this treatment. We can also look at this over time, 24, 48, 72, 96 hours of treatment in whatever cells. So up to 48 hours, everything's alive. And then here's whatever the cells are remaining at 72 and 96 hours. And what we see is that the heterogeneity in MCL1 expression seems to increase. We also see the overall level of MCL1 increasing in the remaining surviving cells over time. And so these types of experiments, it's hard to draw cause and effect relationships. But what we would argue is that there's going to be variable degrees of MCL1 expression and stabilization, and that depending on the level that a cell has when it experiences the MEK inhibitor treatment, and depending on the level that it's able to stabilize, that's going to have a major contribution to its fractional killing, or when it actually dies. And that sort of links in with the MCL1 small molecule inhibition data, because we're saying that no matter what level of MCL1 you have, if you add an inhibitor of that protein, now all the cells are going to die much more synchronously. And we think that those two things are related. If you have more heterogeneous MCL1, you're going to die at different times because you basically have more that you can, hold on to in the cell and can survive with. Another experiment that speaks to this, and I think given the time, because we had some good questions, I'll maybe finish with just this and then some closing comments, was to take our starting population, in this case A549 cells, clone individual cells from that starting single population, allow those clones to grow up into their own populations for a few weeks, and then treat them with compounds and examine their behavior. And so one hypothesis would be that all these expanded clones would behave similarly if there were randomization occurring quickly. Another hypothesis would be that there would be some stable differences, if there's actual different epigenetic states present in the population. And so our data is more consistent with the latter, and it's quite remarkable considering that we have to expand these for 2 or 3 weeks before we start the treatment. But we collected a number of clones, and we're able to find those, including 10 and 16, which consistently would die quite effectively, and other clones like 6 and 15, which were quite tolerant of the inhibitor treatment. And this was not observed only with one MEK inhibitor, it was observed with a second one, but it was distinct for distinct lethal stimuli. So these same subclones, just in different wells, now treated with a proteasome inhibitor, are all very similar in their kinetics, and treated with an inducer of non-apoptotic cell death here, all very similar. So this uniqueness between subclonal populations was very specific for the MEK inhibitor treatment. And we thought that was quite remarkable. And again, speaks to this sort of summary point that whatever the mechanisms are that yield differences in fractional killing, I think they're going to be quite specific to different lethal stimuli. Okay, so then we wanted to relate this back to our model. What we saw was that the clones that had very low sensitivity actually had quite high levels of MCL1 and the pro-apoptotic protein BIM. And the ones that had high sensitivity to dying, they had low levels of MCL1 and low levels of BIM. And so what we think is going on here is that these cells, which are able to survive longer, have a high level of MCL1 that acts as a sponge to basically soak up all these pro-apoptotic BH3 proteins. And now an important point is that we are non-apoptotic people generally, and we are only moving sort of in the reverse direction against the current into apoptosis. There's other pro-apoptotic proteins here we should probably examine, PUMA, for example, being one, which we haven't done. But what we think is that these low sensitivity cells, they're able to buffer the pro-death accumulation of BIM and other proteins. And we have some evidence that this might involve activation of the JNK pathway and direct phosphorylation of MCL1. We present that in the paper, but I think we have a lot more work to do there. Okay, so this is our sort of summary model. where we have cells that have different levels of MCL1 expression. If you have only a little, when you get treated with a MEK inhibitor, you're going to die relatively soon. And if you have a lot, it's going to take you a greater amount of time. But again, I just want to emphasize a couple points. One, when we screen, we screen 10 different non-small cell lung cancer cell lines, many with RAS mutations, just like A549. The response to even MEK inhibitors is highly variable. There's no one fractional killing response, even within linearly related cancer cell lines. Secondly, the role of MCL1, even in the subset of cell lines that die and have nice fractional killing kinetics, MCL1 appears to play a greater or lesser role between cell lines. So now you're getting a really complex picture of what might be regulating fractional killing. The role of MCL1 in regulating this fractional killing in response to MEK inhibitors, that doesn't seem to extend to other lethals. So I showed you some data with proteasome inhibitors, and we looked at other lethal molecules. MCL1 is less important. Well, it's important for some, but it's not universally important as a regulator of fractional killing. In all cases, it's very context specific. The factors that govern fractional killing in other contexts, they don't appear to modulate MEK inhibitor-induced death. So these are things like P53, which is important for DNA damage, ROS accumulation, as I mentioned, cell cycle state. It seems like there's a unique constellation of inputs to fractional killing. But now that we can measure it in a lot of different conditions, I think we can study this. And then, of course, there's other things other than MCL1 that are important. And so I think I'll stop there in the interest of time and have those as my summary points. It doesn't present this a unified picture that I think we'd all love. But I think it just speaks again, maybe on the other hot side, that there is much to do and to really understand these differences between contexts and what might govern fractional killing in those states. So I'll just stop, and I'll thank Darren and Vito and everyone there at the Systems Biology Center again for the invitation. Thank you for the questions during the seminar, and happy to answer any other questions you might have at this time. Thank you.
That was excellent. Thank you, Scott. Yeah, so John has another question for you in the Q&A.
Okay.
Do you want to read it?
Okay.
Or you
see it. No, I got it there. Yes. Okay. Is MCL1 really responsible for the differences, or does it just monitor them somehow? Can you take clones of some cell, modulate the MCL1 levels more or less in each, and reliably observe different sensitivities? Clones of some cell. Right. Right. So these are good questions. We think it actually is functionally quite relevant, if that's part of the question here. I didn't show you all the data that we have. We've done the small molecule inhibitor studies, right, where we have what are really quite potent and specific MCL1 inhibitors, which alter the kinetics. So that's functional data. We've also deleted MCL1 with CRISPR, Cas9 technology, and we see the same effect. So if we delete MCL1 and treat those MCL1 deleted cells with the MEK inhibitors, they now die much more synchronously. The fractional killing is accelerated relative to the unmodified clones that went through the CRISPR process in parallel. So we think that it really is important. I guess a point here, though, is that even those cells that lack MCL1, genetic mutation, it's not as if the cells go from no death to 100% death in an hour. There is still quite a bit of heterogeneity. They still, there's still variability between the first cell in the population to die and the last cell in the population to die. And so now we want to understand what that next level of variability is. And again, it seems like there might be multiple mechanisms. I hope that was partly.
That seemed to be the main gist of the question, is the functional relevance of the MCL1. And your data look like it's strongly supporting that. I have kind of a related question about that, is these clones that have the different levels of expression of MCL1 and the resultant differences in their sensitivity to the MEK inhibitors. Have you tried to, how long have you let them go in culture to see if they regenerate the variability that you see in the initial population? Do they stay sensitive, or do you see some become more or less sensitive over time? This is an open question that we have, is what is the evolvability of these types of states, the sensitivity or insensitivity, and what does that look like? I mean, that's the idea of these essentially discrete functional conditions or states that the cells can jump around in. Have you considered that or looked into that at all?
Right. Like how long, for example, do they sit in an idling state, before they might exit that? we haven't looked at it. And those data were a little bit scary to us because, we isolated the single cells. They grew up for 2 weeks until we could get enough cells to reseed them into 96 well plates and do those assays. And then we froze some, but we didn't carry them out longer than basically 2 or 3 weeks to run that experiment. I think as a follow-up, that is obviously, as you point out, right, that's the big question. How stable are these states? How quickly do they revert? What's the basis for them? We focused on MCL1 because at the time that seemed important. We have no clue about any of those questions, Darren, that you're asking, which are all good ones that we'd like to know. And I think we'd all like to know, right?
Really
again, I just want to emphasize the one point, though, that really shocked us was that those stable states were only specific for the MEK inhibitors and that pathway.
Right.
There was no difference in their sensitivity to other lethal molecules, which I think is really remarkable.
It is, yes, remarkable and frightening at the same time.
Yes. Okay.
Another question?
Yes. Is it from John? Is it possible that some of the remaining heterogeneity is just random? All cells are randomly cycle between states that are more or less sensitive. Yeah, I think there's an element of there's going to be element of different states due to the cell cycle, due to their metabolism, and so on. I mean, we've tried to isolate those, but maybe they have interactions such that when the experiments we did tried to isolate those individual variables separately, maybe you actually need a cell cycle state combined with a metabolic state, and then that puts you into a tolerant condition or a sensitive condition. And we didn't go that route. But I mean, I think whatever processes are going on, I mean, I think we should be able to isolate them, right? I mean, they might be happening stochastically over time, but I hope that one day we can actually figure out what those states are. Okay, and then from a different attendee, with fractional killing in mind, how do we go about treating cancer cells, simultaneously combine drugs or put them in sequence? What order of combination? Right. So Vito and Darren have this really exciting paper on cell systems from, I forget, did that come out 2019 or 2020? I can't remember. Recently, anyway. It was 2019, right? Where they're looking, at how addressing this exact question, right? Like, how do you combine drugs to maximize potency or efficacy, but minimize toxicity? And so for fractional killing, here's a scary example that is always in my mind, because I teach this in my undergrad chemical biology course. There is a species of bee, and bear with me, that cultivates a bacteria to sterilize its own larvae. And what they've found was that this culture that this bee generates to protect its own larvae secretes at least 9 different cytotoxic antibiotics. And so one thought is that the combinations that you might need to address all the different points of resistance or tolerance are so vast that you need combinations of drugs that are of an exceptionally high order, nth order combinations. And I think to this point, there's been lots of work on double combinations, some small amounts of work on triple combinations, but maybe we need to be thinking larger and going up to 5, 6, 7 order combinations. How you translate that into the clinic is difficult, but of course, many of our best therapies are combination therapies that have like 4 different drugs that empirically people have added one after the next and realized are good. So I think we'd like to do it systematically, right? And there's no clear answer to how to do that. But having a method where we can maybe do these combinations is, again, something I think that we're interested in. That was a bit of a ramble, but I hope that helped. Okay, Jason, how much variability do you think exists in the cell's ability to make the fluorescent markers, or the amount of the switch molecule to activate their nuclear localization? Right, so like a technical, does the method actually report on the death, you might say, and then the live cells? We did a lot of characterization in an original paper, and we feel pretty confident that this is the marker is fused to an NLS. It's quite potent. We've looked at some cells. The only limitation we found is that some cells, for whatever reason, for the few hours after they divide, they're reconstituting their nuclei, and they're very slow to concentrate the MCATE in their nuclei and then allow us to detect it. But that's only a rare cell lines here and there. In many cases, we can watch cells in real time divide, and 10 minutes later, they have enough nuclear MCATE that we can count it quite effectively. So there's a little bit of a technical concern there, but since we're measuring hundreds and thousands of cells, potentially in a condition, we have a lot of redundancy. It's not like we're just tracking a single cell.
All right, we are at 1 o'clock. So I would like to thank you again, Scott, for that wonderful presentation. We are now going to try to switch over to the open forum, open discussion for students and postdocs. Any students and postdocs that would like to attend are welcome to join us. And I just posted the link in the chat. You hopefully will be able to just click on that link and it should open your web browser that then gives you the ability to essentially leave this and open it up in the next section. But Scott, I'll give you a 5-minute break if you want to, and I'm going to use it myself as well.
Right.
So we'll close here and switch on over to the discussion starting up at 1:05. All right.
Thank you.
Thanks so much.